Oh, that's good. I used to do that, record lectures and then listen to them. My kids were so funny because I remember my daughter was just old enough to talk and she would remember words like clavicle, mammal, you know. Well, you left on one side. All right, so the recording is stopped. The exams are graded. Um, Take a look at them. I was just looking through them. I just finished grading them this morning. The essays were fine. A couple of you really need to understand this compare and contrast question. That's an important question, especially in science. So if you're not understanding what I mean about the importance of phrasing the compare and contrast question correctly, then by all means come and talk to me. It's really very simple, but it is important that you understand how to do those questions. So those weren't the big thing, but boy, I tell you, the beaker and the pictures and all that, man, you all did pretty well. Something like the cell structure, I'm like, you knew that nobody missed that Professor Clark was a stud, which was nice. But anyway, I'm going to talk more about this in class. So Monday, if I come in and start rattling on about lecture, then do me a favor and just kind of remind me that I do want to go over a couple of these questions. I want to make sure that we understand how this all works. I know it's kind of confusing and there were a lot of numbers, but the important thing in science sometimes is looking through the haze and finding the important part. So that's kind of philosophical, I apologize. So let's do that on Monday, though. Do look over your exams, please. Please look over your exams. I mean, for a variety of reasons. It benefits you if something's marked wrong. It benefits you, obviously, from you learning from, with the kinds of questions. I mean, obviously, I'm not going to ask you the same question again, but I might ask you the same type of question. I had this ecology professor at UCF. I must have taken his class 5 times. By the 5th time, it was the easiest class I ever took. It was community ecosystems or something like that, some graduate course. But I knew what he was going to ask on exams. I just knew. I knew exactly how to study for this guy. Makes sense, right? All right, so take a look at those. Chapter 7, now we start on some new stuff. Now it really gets kind of hairy. It's interesting, but you really have to follow along with me because it's very easy to get lost. So what we're going to do in Chapter 7 is we're going to explain that picture that you see on the slide, right? Look at all. What that is basically is a chemical schematic. Excuse me, it's like an electronic schematic, but it's a chemical schematic, so that each dot represents a substrate, and each other dot represents a product, or maybe the substrate for the next one. And if you look in I know that the resolution is great, but you notice that we're all talking about metabolism. These are metabolic pathways. So what this is a chemical map or chemical schematic of the way your body goes through the process of metabolism, depending on what they're using, right? Like lipids, that's over here on the left-hand side. Carbohydrates, that's where we're going to focus, actually. That blue that is sort of moving its way down the middle of this picture, this stuff right in here, that's where we're going to focus. Carbohydrates, this is how you metabolize nucleotides, nucleic acids, and all this sort of stuff. You can see there are even some amino acid metabolism, vitamins, secondary metabolites, how you make things. Anyway, that whole balance. So this is kind of the wiring of your inner balance, that homeostasis in your body, right? Keeping everything in balance. So we're going to spend our time in Chapter 7 going over that. Thank goodness we're not. Thank goodness we're not, because this is an awful lot of information, isn't it? Imagine an exam. Give me in your essay, describe all the different ways, metabolic pathways, and how they occur. You all would shoot me. And you should. But we are going to look at the one in the middle, the carbohydrate. Now, the way they happen with the other material, other substances on this is also important, but let's just focus on the carbohydrate one in Chapter 7, and we'll leave all the other ones for honestly advanced kind of biochemistry classes. And the reason why we're going to do the carbohydrate metabolism is that is actually in cells, that's the preferred way to convert energy. Remember, nobody makes energy, but the preferred way to convert energy, one form into another, is by doing it using carbohydrates. Now, your body will use these other things, of course, lipids and all these other things, but carbohydrates is what we say the preferred method by which it's done. Now, let's step back for a second. I was talking about my community ecology professor, so he would like to look at something like this. If we look at an ecosystem, it's all this big cycle, right? It's so interesting to me how when that homeostasis thing happens at the cellular level, the organismal level, at the species level, it's so interesting how the whole thing of balance, even at the chemical, even at the astronomical level, it's all about balance. Now, down here in the ecosystem, the balance that we got that's going on is this idea of respiration. That's the bottom part. There, you might recognize that organelle there. Remember, that's the mitochondria. The mitochondria is very important in this process of respiration that we're going to talk about. Now, we talked about respiration, a long time ago when the semester started, and we talked about the fact that respiration is a little more than just going, right? Respiration is the whole chemical thing that happens. The reason why we go is to get oxygen, right? We need that oxygen, and we're going to find out when we're done today. Notice there it is. And I bet you my pen's not going to be nice to me. No, of course not. There it is, oxygen on the right-hand side. Now, that is specifically atmospheric oxygen, the stuff you breathe, as I just said. It's not some chemically altered, chemically rearranged. It's plain old everyday atmospheric oxygen. And you're going to find out when we're done with this chapter that oxygen's role in keeping us alive, it's really, 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 really important role, is I don't know, anticlimactic. It's kind of a weird thing. You think about, it's all driven by oxygen. Well, it is kind of driven that way, but it's sort of in a way that you may not expect. All right, so we'll get to that. So we'll do the bottom part first. We'll take this, and so what we're going to basically do with the bottom part is we're going to take in the mitochondria. Eventually, we'll details on that later. But we're going to take food. Okay, that's what organic molecules are. Organic molecules are food. That's just a fancy way. Organic molecules, carbon and hydrogen, carbs, lipids, things we eat, okay? So we take those organic molecules and partially in the mitochondria, not all of it, but a big chunk of it anyway, is going to use that energy in food. Okay, now I'm going to use the words that we talked about in, I don't know, chapter 6, metabolism, or 5, whatever they were. So this arrow that I'm drawing right here is the process of oxidizing organic materials. Now, I want to jump back and forth between these slides, all right? So don't freak out on me there. All right, remember that process of oxidation. So this is a little bit blast to the past, right? When something is oxidized, like sodium in this picture at the top that we play with, and down below C6H12O6, that's glucose, food, right? If you're into balancing equations, fine. If you're not, don't worry about what the numbers mean in front of the atoms. You probably know what that means, but this ain't a chem class. Anyway, when sodium becomes oxidized, it loses an electron. Yes, that's the physical. That's the physical. What also happens in that process is sodium transfers. I was going to say loses energy, but that's not really the good word because energy is never lost. Just like an electron, when something's been reduced, it's got to have someplace to go. The energy is being transferred, you see. So it's losing technically from sodium, but it's now the energy is being transferred in this particular case to chlorine. If you want to follow the transfer of energy, follow the movement of electrons. It's all about the electrons. They're the energy-rich part of the atom. Okay, they're the part that hold that energy that we're actually doing that whole process of eating food. See down below, same thing, right? So we take sugar or starch. Starch is a polymer of sugar, so you can chop it up, whatever. And you take other things, yes, the lipids and all that. But I'm saying, we say in metabolism that this little glucose nugget, C6H12O6, it's one of those little hexagon-shaped things that we saw a long time ago. They were colored green. He's excited. Anyway, that is the sugar of choice. So it's a glucose monomer, right? And it becomes oxidized by a very good oxidizer, by the way. See that right there? Where's that come from again? So the food in our body, when we're doing this oxidation process, where's that O2 come from? It's actually 12 O2s, but again, don't worry about that. That's me sighing because you're not answering. There we go. It's in the air, right? It's about 20% when we used our probes the other day, so it's a very good oxidizer. So it strips electrons off of glucose, right? And in this particular case, we'll do a little bit of alphabet soup, and I'll explain for reasons why, but just kind of follow me on this. When the electrons come off of glucose, C6H12O6, so do the hydrogens. They don't come off directly, so more on that later on. But anyway, back to the energy part. So the energy is being transferred from the glucose, okay? And that energy that's being transferred from the glucose is this energy right here. Okay, now that energy, just to make the connection, is the energy. Why did it get dark in here? I always think I'm getting glaucoma. All right, that's the energy that does this. Let me write it out. And hopefully by now you're going, I get it, because this is an endergonic reaction, is it not? I'm building a bigger molecule. It's very simple. I got an ADP, it has 2 phosphates, and I'm putting a third one on there, aren't I? I'm making ATP. We know what ATP is. That's energy. It's food. It's currency. It's stuff you make. And then we use that ATP, over here. We do this when we're doing things like climbing stairs or needing cellular work, right? So that energy there in this whole process down below is not being given off just bloop. I mean, it is. Some of it is. Not everything's efficient. Every time this happens, by the way, not 100% of this energy actually goes to making ATP. A lot of it is lost, especially in us mammals, because we use a lot of it to keep ourselves warm. Not everybody stays warm, right? Who doesn't keep themselves warm? Did we talk about this already? My routine is getting old. 100%, right? It's not 100%. Never. Why do we keep ourselves warm? Us, mammals, why do we keep ourselves warm? You do know the answer if you think, oh, I have no idea. But you do. You know what happens when you heat things up. And you knew, though, that living things are chemical reactions. We're just fancy chemical reactions. We're very delicate chemical reactions that do this dance. We're really no different than what goes on in a black hole or a collapsing star. It's all this chemical dance that occurs. But we keep ourselves warm, right? Us mammals keep ourselves warm. What happens if you keep that chemical environment where those chemical reactions are occurring? What happens as it gets warmer? What happens to those chemical reactions? We just did the lab on it. It didn't work out exactly that way, but we certainly have talked about it enough. So more reactions occur, the probability increases. Okay, so heating up a body in the case of a mammal, and birds, by the way, doing that allows you to do chemical reactions, fancy chemical reactions, like mammals have fancier lives. We have living young, and we give birth to young, and all this other kind of stuff. And many of us mammals are social creatures, blah, blah, blah. But you can do that kind of stuff. And birds are a little weird that way, too. They have very social interactions. If you're a cold-blooded critter, like a reptile and a snake and fish and all that, your metabolic rate is a lot lower. Or that's one thing. You're not going to have that extra heat to speed up those reactions. So you're really sort of governed by the environment. That's why lizards and snakes are, when I was a kid, they were easier to catch when it was cooler outside. The reactions are slowing down, right? All right. So you understand all that. But the advantage of being a cold-blooded critter, by the way, is the fact that you don't have to eat as much. Snakes can, average-sized boa for whatever could eat every 3 weeks, let's say, right? You can't go 3 weeks. But none of that energy transfer, regardless of whether you are cold-blooded or warm-blooded, is 100%. I mentioned about the car. A gallon of gas, it's only about 30%. All right, but that's off on a tangent. Anyway, now back to the equation for a second before I move back to the previous slide. I want you to follow the bouncing letters again for me, because we need to know the inputs and the outputs of this kind of reaction, and you're going to see it again, for example. So C6H12O6 and an oxygen. Again, don't worry about the balancing. Let's just follow the bouncing letters. CHO, which is the glucose on the left, I'm just ignoring the numbers of atoms, is having its H removed. CHO is removing an H. So very simply, if you take CHO and take out the H, you got CO. Right? You got carbon dioxide. And O, if you take the H, and it's got to have some place to go, so if something's been oxidized, something's been reduced, the O, in this particular case, that's where the H goes. So the H goes onto the O and makes water. Now, this is really water, not the water you drink. This is what we call metabolic water. It is the water you drink in chemical sense, but it's not water you get from drinking. It's a little bit of water you get from this whole metabolic process. You don't get a lot, not enough to keep you alive. You have to bring in other water. Now, are some critters, some desert critters, desert mammals, there are some organisms that do this, that do live on that metabolic water, or live a lot on that metabolic water for reasons because they live in the desert, right? There's a desert in Africa where it rains once every 10 years. It's amazing. And there's fish that live out there too. No, it's in southern It's a smaller, not the, it's part of the Kalahari, I think. Is the Gobi in Africa or is the Gobi in Asia? I don't remember. Yeah, me neither. But it's a part of the Kalahari, which is the southern desert in Africa. And there's a fish that lives out there. It lays eggs when it's rainy, when it rains and there's puddles, so it rains for a while. And then the eggs go down into the sand and they basically hibernate for another 10 years until it rains again. What, for saying the desert in Asia when I was talking about Africa? Now, the other side of this equation, this is where my college ecosystem professor would appreciate this, is when we look at this in terms of an ecosystem, then we get that cycle again, the big old cycle. Because here's the carbon dioxide and the water I was just talking about. The water is a little bit misleading. You know, this is that metabolic water, okay? You don't make enough of that, and probably whatever you make, you use, to be honest. But nonetheless, water. And in this particular case, it's going to be rain, right? Because what we're talking about at the top is photosynthesis, right? That process by which plants convert, not make. They convert light energy from the sun, the ultimate source of energy on the earth. Everybody says is always the sun. Whether you're a herbivore or a carnivore, trace it back, it's always going to be the sun. So the sun drives this cycle we're looking at. And we'll do photosynthesis in, what, chapter 8, right? Next chapter. All right, so you take light energy and you convert that light energy into organic molecules. Now, remember what that organic molecule was in the previous one. And again, I'm not going to go with the numbers, and I apologize because I do my mouse with my right hand, but I'm left-handed. It's like when I played soccer, same way. But anyway, that organic molecule is this thing. It's a CHO, C6H12O6 specifically. So what we basically do in photosynthesis, now we're going ahead in time, right? Just a little bit. In the next chapter, we're going to learn how you take carbon dioxide, CO, and HO, or H2O in this case, and put the H onto the CO. So the opposite, bingo, the opposite of what happens in respiration. So the real nice thing about this is once you understand the chemical equation for respiration down below, you simply reverse the arrow and you got yourself photosynthesis. This is what plants are doing. They're doing it this way. Now the only difference between this right here, this process is a catabolic reaction. How do I know that? Well, a big molecule is being torn down. Glucose is being torn apart. Okay? So this is a catabolic reaction. And catabolic reactions release energy. They're exergonic. If you want to make something, if you want to go the other way with the arrow, take carbon dioxide and water, and you want to make something more complex, the oxygen's not, I get that, I understand that, but if you want to make something more complex like glucose there, right, that's a very complex molecule, it is an endergonic reaction. That's why you need this on the right-hand side. And in the case of us, in the case of plants, it's this thing. I'd switch over to the yellow real quick, but or I could do that. Anyway, y'all get the idea, right? It's the sun. There we go. I draw like a third grader. Anyway, so the energy from the sun drives the reaction the other way because it is an endergonic reaction. You take the energy that's stored in the chemical food, C6H12O6 glucose, and you release it. It's like a battery. It's releasing that energy, and you're releasing that energy to make ATPs. And then we don't go into it in this class, but somewhere down the road, you know, if you took an A&P or something, but somewhere down the road, that ATP, someone's going to show you, well, I do a little bit here, like when we talked about the sodium-potassium pump. It's a nugget of energy, if you want to think of it that way. That ATP goes throughout your body and does whatever your body needs for energy. That's what we call the currency. Do I need to spend any more time on these slides? We all cool with this? We understand how that great cycle works. I feel like you're singing the tune from Lion King. The cycle of life. We'll talk about photosynthesis next. Very cool. See, it's all chemical. Now, what did I say was going on in glucose? Back to that again. The hydrogen is being stripped off of or in respiration, excuse me. The hydrogen is being stripped off the glucose, right? It's being removed, so this oxidation is occurring, it's being put onto the CO. I want to focus a little bit now about the second part of the process, where the hydrogen gets stuck onto water. So now I want to look at the process by which hydrogen, H2, All right, now where's the H2 coming from? Get rid of the H. Scrap that. I want to talk about where the H in the glucose on the left. I want to talk about the process when this occurs. The H plus the O, again, this O, this H, and then, you know, some balancing, whatever. In other words, the water, the metabolic water. I want to focus on that process. Now, I said the metabolic water is a little tiny bit of water, not all that important in terms of water needs, but what I want to focus on, what happens in that chemical reaction when you put those two together. Because for one thing, if you think about this, there's a lot of oxygen in the atmosphere. What were our probes were saying, what, 20%? And hydrogen is common as dirt in the environment. It's everywhere. I think 99.9% of all the elements in the universe are hydrogen. Isn't that amazing? So 0.1% is all the other elements. Hydrogen is everywhere out there in the environment. So think about that for a second. If there were a way in chemistry, if there were a chemically that we could in a factory take this hydrogen, which is, gosh, everywhere, right? If we take that hydrogen and we could mix it with oxygen, which at least on Earth is everywhere, we could make H2O, couldn't we? We could make water. Water shortages, some areas of the world don't get as much water, others do, all those things that you've heard about. But the point is, wouldn't this be great if we could do that? Both things common as dirt, at least oxygen is common as dirt on Earth. Put them in a lab, stick the two things together, make yourself water, put that in a tub, and nobody has any water problems. Maybe we could make this portable, and you could have it in your house. There is a problem, though. There's a very big problem. When you allow hydrogen and oxygen, up in the upper left right there, free energy, the fact what they're showing you is energy is being released here. When you allow free hydrogen and free oxygen to freely mix by themselves, they do very nicely and they make water, but it's not in a controlled fashion. It's what we call an explosive release, and you may or may not remember that that's how the part of the shuttle, the main tank of the shuttle, not the solid rocket boosters on the side, but the main tank was split and it had hydrogen in one, oxygen in the other, and at the back of that they mixed them, which exploded, caused thrust, and et cetera. That's why Challenger blew up. Challenger blew up because what happened is an erosion occurred between those two tanks because of one of those solid rocket boosters, and what you see on the left-hand side happened. It was an explosive release of energy. Now, in the back of the shuttle, they were controlling it. It was still a pretty good release of energy, but it's kind of like in your car. Putting a spark plug, putting an igniter and gas together in your garage, a gallon of gas sitting in a bucket and throwing a lighter or throwing a match or whatever, it's going to get a release of energy. It's going to get an explosive release of energy. Kaboom! You wouldn't do that. But if you could somehow release that energy a little bit of time, like you do in the engine of your car, right? Just a little bit of squirt, little igniter, boom, boom. We have little tiny mini reactions, enough to push a piston. If you could release that energy in a more, say, controlled fashion, it would be more usable, right? And that's what cells are doing. So on the right-hand side, you see what happens in cellular respiration. Now, the oxygen is the same thing on the right. So let's do, that's the easy part. That's atmospheric oxygen. Okay, don't worry about the half. You know, again, that's balancing. But what happens to the hydrogens is what's important here. If you take a look at those hydrogens, they become split. So the hydrogens, in this case, there are two of them. That's why the two get split into their proton and their electron, right? So remember, on the left is called a hydrogen ion, or it's called a proton, because in this particular case, that is what it is. Hydrogen is very simple. It's only got one proton and electron. So if you strip it off, you got a proton. Now, the hydrogens, you follow the arrow, they eventually reunite with those electrons, and we'll talk more about that later on. But again, the important part is the electrons. Look at those electrons. Those electrons have energy, all right? They have energy in which they can transfer. And what those electrons do is they eventually reunite with the hydrogen. So they all come together at the end, the H pluses, the electrons, which are minus, the oxygens, and they make that water, just like the one on the left, but slowly the energy is released from the electron. So the electron releases its energy. Okay, now I'm going to do a big picture thing for a second. This is the energy the electron has by virtue of being in a glucose molecule, which is an energy-rich molecule. okay? The glucose molecule has the energy by virtue of photosynthesis, and the energy from that is from the sun. So bloop, 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 bloop. Now, the energy is being released in a controlled fashion from the electrons on the right, and we can control the production of ATP. Not boom, but boom, 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 making ATP along the way. See, that's what those little, what the hell those things are, little squiggly things you're trying to understand. I tell my students, if you were the top of a 10-story building and you wanted to get out, there would be 2 ways to do it. I'm not counting an elevator. You could jump out the window or walk down the stairs. Both of them will get you to the same spot. Both of them will release the same amount of energy. If you jump out the window, when you hit the ground, bam, that's a release of energy. That's why it hurts. It releases it into your body. When you step off of a stair step going downstairs and your foot hits that next stair step, that's a release of energy too. That's your body hitting against something. Slowly, slowly. So you walk down 10 stairs, slowly, 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 slowly. A little bit of energy being released at a time. You get to the bottom. You're not all mashed up on the sidewalk because you didn't release the energy like explosively on the left-hand side. You release the energy from the electron as if it's walking downstairs. Little bit of it. And then at the end, you let everything happen. Now, the reason why you don't have an explosion in the end, and the reason why when we make metabolic water, we don't explode, is because by then, the hydrogens, all of the stuff that had energy is low. See on the left-hand side, it's very low energy because that energy has been transferred to the ADPs to make more ATPs. So at the very end, that reaction where it typically would be explosive is not. It's very anticlimactic. You breathe in oxygen and the low energy electrons and the hydrogens all get back together and they make water and everything's fine. We don't explode. Nobody's exploding. Make sense? So we're going to follow the bouncing electron here for the most part of this. There's going to be a lot of other things we're going to talk about, but it's going to really have a lot to do with this electron. Are we cool with all that? You understand how that works? A little bit of time, release that energy. Bloop, bloop, bloop. Nice. Now, we need something to move the electron along. In other words, in respiration, we need something that helps move the electron from the food eventually to the water. Eventually to the water. We haven't talked about this before. We've talked about oxidation always involves reduction and vice versa. But in the case, in many metabolic pathways, that whole oxidation and reduction process is going to be helped along by something we call a carrier, specifically an electron carrier. And electron carriers do exactly what the name says. They carry electrons. They don't keep them. They don't use them. They specifically or they take an electron from point A to point B. From substance A to substance B. All right, so we've been talking about electrons here. Let's talk about who carries those electrons. And then what I'll do eventually is I'll put all these characters together in a big picture for you, right? So we've got high energy electrons. We have something that carries them. Now, the names are intimidating enough. A nicotinamide adenine dinucleotide and flavin. David, that probably looks familiar to you because that's riboflavin and all that sort of stuff. Flavin, adenine, dinucleotide. Let's go with NAD and FAD. How about that? Let's go with NAD and FAD only because I tongue twist myself all over the place if I use them. And in the picture down below, I show you a NAD with a plus and a NAD with an H. And you'll notice right below that, I've written something, a word you should recognize. When we see NAD in the plus form, we know that it's been oxidized. Don't really care too much about the chemical equation down below, but if you really, that's supposed to be a question mark, holy cow, Steve. But if you really want to know what's happening is that other hydrogen gets stuck up here, see? All right, so anyway, the point of fact is, and I really sort of summarized it in this red box up here, it's really important that you understand when NAD is oxidized or reduced. I don't care about you looking at the chemical structure, the picture you see there. It's very simple. If it's NAD+, it's oxidized, so it's not carrying an electron. And if it's NADH, it's reduced, so it is carrying an electron. Okay? Same thing with FAD, which is another electron carrier that we'll meet a little bit later on. It's not as prominent as NAD. Now, the NAD+, plus 2 hydrogens, is an NADH and a hydrogen. All part of the balancing. Don't get real hung up on that. Oxidation and reduction is the key thing here. FAD is oxidized. FADH2 is reduced. So if the H is on the NAD plus or the FAD, then it has been reduced. See, the H's are the protons that are going along with those electrons. Remember I said it's this play that's occurring between the hydrogens and the electrons that are in glucose. Okay? Big takeaway here, if you see these again, understand if it's being oxidized or reduced. And don't worry, I'll say it over and over and over again. So you'll understand. All right, step back for just a second like we just walked into the mall, that you are here kind of picture, and let's see where we are in the cell. The little squiggly thing that you see on the right-hand side that looks like a bean, that's the mitochondria. And you'll notice right away that not everything that we're going to talk about, and yes, before you take another swig of coffee, we are going to talk about all these things. They're not quite as intimidating as they look. But all these things we talk about, not all of them happen in the mitochondria. They only happen in the mitochondria past here. See, out here, the cytosol, remember that's another word for cytoplasm. So this is the cell, and out in the cytoplasm of your cells, we're already doing respiration, and we've been talking about the mitochondria. The mitochondria just made respiration real efficient. It didn't invent respiration. You see, this first part that we're going to talk about out here in the green, glycolysis, that literally means sugar splitting. Glyco is sugar, and lysis is to split, right? And when you split something, when you catabolize something like glucose, you release energy. Catabolism, exergonic, right? Anabolism, endergonic, anabolism. And just remember those always. So if you look down below, glucose is being catabolized, torn apart, split into pyruvate. Oh, there's that pyruvate dehydrogenase thing. Remember that enzyme we talked about? So here, pyruvate comes back. We'll talk more about that too. But notice down below what's happening. Some ATP is being formed. So because you glycolized glucose, because you chopped it, because you catabolized it, what you did is you released some energy. And then energy was used to make ATP. And it was used to make ATP, because there's two ways you can do it, in a very simple process. Notice down below, two ways. Substrate level, and then the one we'll talk about at the end is called oxidative. I'm going to come back to this picture because I still need to finish up talking about the electrons, but let's go look at substrate level ATP formation first. Very simple. You already know how it works, just like that. ADP is a substrate, some green thing, I don't even remember what it's called. It's some molecule that has a phosphate on it is another substrate. They bind to an enzyme because they stick together, and voila, wang, ding, ding, ding. What you get is the phosphate ends up on the ADP, and you get a product, okay? Enzyme substrate complex, enzyme product. Nothing different there. Very simple. That's how the ATPs are made out in the cytoplasm of the cell when you split sugar. No questions on that, right? We're cool because that's just your typical enzymatic reaction. Very simple. Oxidative phosphorylation, very much different. I had a student in my class years ago, deaf, and so they had a deaf sign interpreter, and I remember the poor woman trying to, there's no sign language for oxidative phosphorylation. I remember her just doing this with her fingers trying to spell it. So she would ask me to slow down. I can understand. All right, any questions about that? That's how we make ATP out there. Now at the top is something that we'll have to come back to. At the top, you'll notice that what's going on is NAD plus is oxidizing stuff. See, because it says electrons via NADH. That means I know that NAD has electrons on it because it tells me that it is. And so that means I know that NAD over here, my pen would work, but it won't. NAD must have come in, the plus one, and it must have taken away those electrons. And by definition, that means it oxidized something. That means it catabolized something. Blah, 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 blah. Same words. Right? So NAD went in. NADH came out. Now, what happens to the NADH, specifically the electrons on it, because remember, it's just a carrier. You don't really, it doesn't keep it. Like if you fly on a plane, they call those carriers. You fly to Philly, you got to get off the plane when you get there, right? You're not staying on the plane. But we will talk about those electrons later on. I want to go back to glycolysis and look at that in a little more depth now. All right, as I said, the way ATP made is very simple, and it's out in the cytoplasm of the cell. We're not in the mitochondria yet. So let's take a look at the steps of glycolysis. Easy, right? Easy to memorize all those steps. Glucose is by the enzyme. Notice there's an enzyme there, right? Hexokinase. I'm looking at the #1 spot in the upper left. That's an enzyme. Glucose and hexokinase and energy. Oh, what? I thought that this whole process made energy. Oh, God, I love Mother Nature. Priming the pump, folks. Notice at the top, and I highlighted in red, the first half of the process of glycolysis. No, first thing also, too, the sugar is indeed split. See? But the first part is called the energy investment phase. You remember back when we looked at enzymes, I used the word prime the pump? Got to get up over that hill? That's what we're doing right here. We're priming the pump. Glycolysis starts by adding energy. It's called the energy investment phase. It actually takes 2 ATPs to go from glucose to those things that are split. Now, just to keep you, from jumping off the ledge here, I'm not going to ask you to memorize the names of these molecules and these enzymes. God, no. Here's what I want you to know about this, though. First of all, that you invest 2 ATPs. The sugar is split. Enzymes are involved, because I might ask you questions about what happens as heat goes up to the process of glycolysis, or I don't know, if you take glycolysis and put it in an acid that it's not used to, all those things that should trigger protein denaturing in you. So down below now we're following 2 molecules, okay? Well, actually, I'm sorry. At the end, you end up with 2 molecules. Then there's an enzyme that actually converts one into the other. That's what the little arrows are. So down below, we're starting with one of those, G3P. See, when you split glucose, you get a good chunk, G3P, and you get a chunk you have to change. Don't worry about why the arrows are back and forth. It all has to do with balance. So G3P is what you want. It comes down here. All right, now I'm going to use some words that we've used before. What we're going to do to G3P, okay, is we're going to oxidize it. NAD is going to come in and take electrons off of it. Now, the hydrogens are going to come off too, and they'll come back together eventually, and we'll talk about that, okay? But in this particular process right here, NAD plus, and it's in brown, is turned into NADH. So we now have a loaded, again, the 2 just says how many there are. We have a loaded electron carrier. It's going to take that electron somewhere. Notice in this picture there, it's not taking it anywhere. All we've got here is oxidation. Oxidation always has to have reduction. So this picture is only showing half what's going on because nothing's being reduced. Now, notice down below about the ATPs. Sugar was split, all right? Each of those glucose 3-phosphates is going to generate 4 ATPs as it's being turned into pyruvate. So what do you need to know here? You need to know that in the energy payoff phase, number 1, you gather some electrons. Sometimes they even call it electron harvesting, like farmers do, right? So we're gathering some electrons in the NADH part, and we're returning twice as many ATPs as we made. 2 go in at the top, 4 come out at the bottom. So it's important you understand that because it talks about enzymatic reactions, the energy of activation is 2 ATPs, but the energy that you get out of that priming of the pump is 4. That makes total sense, doesn't it? If you invested 2 and got out 2, if you invested 2 and got out 0, living systems couldn't survive. You're going to have to get out more than you need. Now, there are some critters out there who don't have mitochondria. This is how they live. They live simply by just doing this. They take glucose in their cytoplasm because they don't have mitochondria, and they split it, and they live off the 4 ATPs. Now, we're not talking about critters walking around doing active things. These are many times prokaryotes. But if you're into a low energy lifestyle, but that works. You can do that. You do this too, by the way, so we don't confuse. You have a cytoplasm. You do this too, but you do things in addition to this. Remember I showed you the mall roadmap. There were all kinds of other things happening after glycolysis, but they were occurring in the mitochondria. So you do this too. You get 2 ATPs. Well, you get 4, but you net 2 ATPs every time you take a sugar molecule, glucose, and break it down, all right? Any questions on this? What am I supposed to know? What am I not supposed to know? Anything like that? Because there's a boatload going on. Mostly think about inputs and outputs, okay? Think about inputs and outputs, glucose in, pyruvate out. The ADP goes in, the ATP comes out. The NAD goes in, the NADH goes out. And then in the case of the ATPs, know the number. Got it 2 to start, and you get 4 at the end. So you got to know gross yield and net yield, right? You good? Now it gets interesting. See, so the ATPs go off and do their thing. Right? Bloop, 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 bloop. They go off and do their thing, climb stairs, whatever they're going to do. But we still have to resolve this issue here. Oxidation has to follow reduction. Always, always, always. If something is reduced, something is oxidized. If something is oxidized, something is reduced. The electron can't be given away. It's got to have some place to go. Now, NAD is just the carrier. Right? NAD is just the carrier. NAD is just moving it from point A to point B. But now we're going to talk a little bit about where that electron is going to go. And the first place we're going to talk about where it goes is if there is no oxygen present. There are organisms out there that engage in respiration that's known as, I got to spell this because it'll take me forever to write it out, anaerobic. You might notice on the page aerobic. Aerobic means when oxygen is present. Anaerobic, the AN means the opposite, means when there isn't. There are areas on Earth down in the mud, bottom of the ocean, bottom of really deep lakes, especially during certain times of the year, that can become, here's another word for you, anoxic, without oxygen. All right, there are some organisms on Earth that actually die in them. Remember, oxygen is not kind. It's a big electron pig, man. It loves to strip electrons off of things. And there are some critters that never learn to deal with that. So they have been relegated to living in those very specialized, very rare, I guess, kind of, relatively speaking, anaerobic environments. Bottom of the ocean, down in the mud, right? Oxygen kills them. But there's a problem. Oxidation follows reduction. Reduction follows oxidation. They've got to do something with that electron. If you think about it as kind of a plumbing thing, if you don't dump that electron off somewhere, get rid of my scribbling. If you don't, in other words, if you don't oxidized NADH down the road somewhere, if you don't take the electron off, then you don't have any of this, right? Because if you don't oxidize NADH, you're not going to get NAD+. And if you don't have any NAD+, you can't do the reaction that you see in front of you. And if you can't do the reaction that you see in front of you, you never get the ATPs. So it becomes a negative reaction. Putting in ATPs, nothing coming out. Nobody's going to do that. If you do, you're going to die. So you got to put those, you got to dump those electrons off somewhere, right? If you don't dump those electrons off somewhere, all this is going to back up. And if you don't have oxygen around, which loves electrons, you got to find other places for it to go. So we're going to talk now about what happens if you go to the left in the screen here. So here we are in the cytoplasm. There's the mitochondria. We're still out in the cytoplasm. Just done glycolysis. We brought in glucose and we've split it. We'll go this in just a second. We're going to go here now. All right, if you don't have oxygen to take those electrons, you have some choices. If, for example, your yeast cell, yeast is actually a fungus, it's not a plant or anything like that at all. But in the case of yeast cells, what yeast cells can do is when they split glucose and they get their 2 ATPs, which is what they want, so we've really whittled the process of, this is kind of what I'm looking at for you to know for glucose, by the way, or glycolysis. There you got your pyruvates, all right? Now, then another reaction occurs in which the pyruvates lose a little bit of carbon dioxide. But the point is you get something called acetaldehyde. That's where the electrons go. It's almost like a perpetual motion type machine. The electrons, and notice you can see NAD+, the electrons that were oxidized off of whenever that happened in glycolysis, about halfway through, those electrons that had to have a place to go, oxidation follows reduction, go on to acetaldehyde. And now you got a source of NAD+. So those electrons transferring to acetaldehyde, NADH is being oxidized, acetaldehyde is being reduced into ethanol. That's how you make beer. This is what we call alcohol fermentation. In alcohol fermentation, the electrons that were stripped off during glycolysis are put onto a derivative pyruvate, not pyruvate. Pyruvate gets turned into acetaldehyde. That's the carbon dioxide being given off. When folks brew beer, right, and they use yeast, this is what yeast does in an anaerobic environment, it's important that the beer brewing process be kept air-free, because if oxygen is around, it's going to take the electrons. See, the electron's got to go somewhere. Acetaldehyde will take them here, but if oxygen is around, forget it. Oxygen gets them. And they know that, and they also know that in that air-free environment, that at the end of that process, what they get is ethanol. If you let air into it, you don't get ethanol. I used to know this guy. They used to do his home brewing. He was all very into keeping the oxygen out of there. And carbon dioxide is given off. As a matter of fact, Anheuser-Busch, I used to work for them at SeaWorld. They sell off the carbon dioxide in the brewing process. People use it for carbonating drinks. That's right. All right, so that's a yeast cell. That's called alcoholic fermentation. Now, jump down below before you go to the picture on the right. There are other places those electrons can go, but if they go into something other than acetaldehyde, then we call it anaerobic respiration. Okay, just so you're not confused. Alcoholic fermentation is a type of anaerobic respiration, but it specifically involves acetaldehyde taking the electrons. If sulfates or nitrates or sulfur, if something else, that's what's happening down at the smoker vents, if some other material accepts those electrons other than acetaldehyde, then we call it anaerobic respiration. We don't call it fermentation. Fermentation is a type of anaerobic respiration. It's all about where the electrons go. You get it? Regardless of where they go, they gotta go somewhere. Now, animals, we do something like this too. We do it during times of heavy physical stress. We do it during those times when we need a lot of ATP real fast, like danger. We're walking down a path and Mama Bear's pissed off, comes running after us. Boy, we better start running, okay? ATP is, I told you, is volatile. It doesn't stay around very long. It's very unstable. It's ready to break apart because you want it that way because it easily breaks apart and releases energy. It's like milk. It only lasts a couple of days or so before it goes bad, if you will. You don't store up ATP. You only have a few minutes worth. So you're running away from Mama Bear. You're probably running away fast. You're probably running longer than 2 minutes. Hopefully you're running faster. What happens? Do you run out of ATP and you stop and become bear food? No. One of the things that'll happen is your body in the cells can switch over to something called lactic acid fermentation. Cool story. Cool story because it just shows how Mother Nature, that balance thing. All right, so in lactic acid fermentation, it's easier. Basically, the electrons have to have some place to go, and where they go is right back on the pyruvic. So there's not the acetaldehyde part. Here's the problem. The byproduct is lactate. Your body does nothing with lactate. It metabolizes it, right? Tears it down eventually, but your body doesn't eat it, doesn't use it, doesn't do anything. So you can't hold your breath and get drunk if you're thinking you can do alcohol fermentation, right? It's not going to work. Because what happens when we don't bring in oxygen, there's times when we're trying to make ATP as fast as we can, two's better than nothing. Making 2 ATPs is better than making a whole bunch, which we'll see how it happens in just a second, because, man, you're trying to get away from the bear. But the clock is ticking. The reason why the clock is ticking is because this is forming. Every time you do this process, you're acidifying your cells. Every time you do this lactic acid fermentation, right? It's fermentation, again, it's anaerobic respiration, and in this case, in animals, fermentation, the acceptor is pyruvate. Remember, in yeast cells, it was acetaldehyde. That's, again, very different than anaerobic respiration in general. All right, aerobic respiration, just so you know, is oxygen is the acceptor. All right, so right here. So any place the electrons go, if it's not oxygen, it's called anaerobic. All right, so now the lactic acid builds up in the cells of your body, and you're trying to run away from the bear, and you keep running, you keep running, keep running. You need ATPs, man. You run out of ATPs, you're bear food. What happens to the pH of your cells? What happens to the pH of your cells when you engage in lactic acid fermentation, or you make lactate? And what happens when things become too acidic around proteins? Or I guess I could reverse the question, but either way, what happens when proteins become too acidic? Yeah, and are there proteins important for glycolysis? Let's see. Oh my God, I'll say. There's one, there's one, there's one. Every one I'm underlining is an enzyme, and enzymes are all proteins, aren't they? So that's what I mean about not knowing the names of them, but perhaps understanding the significance of them. Yeah, absolutely. Those proteins are going to become denatured. You're going to start denature the proteins. They're going to stop working, and you're going to become bear food. If you do escape from the bear, Okay? We say you pay back oxygen debt, just like a money. That's why you're increasing the oxygen levels in your body by breathing heavy like that, because you're trying to metabolize that lactic acid, among other things, trying to get rid of it. Try and tear it down. You don't want it sitting in your lungs. It's going to become if you ever watched A Few Good Men with Jack Nicholson, that's what the kid dies of. He dies of lactic acid fermentation in his lungs. Anyway, if you haven't seen the movie, it's a good movie. I eat breakfast every morning. Anyway. All right, let's talk about the mitochondria for a second. Here's a picture of your book. I thought it was a particularly good one, so I put it in there. Mitochondria is a very interesting organelle, because remember, it's one of those organelles that it might have been living by itself. It may have been a descendant of some free-swimming, free-living proteobacteria billions of years ago. The reason why we say that is because it's got two membranes. It's a membrane within a membrane. There's the intermembrane space. That's a bilayer phospholipids with proteins. You've heard that before. Oh, yeah. Yeah, as long as you could keep, I'm thinking, could you make enough ATPs, though? Only 2 at a time. After a while, maybe you might be able to condition your body to live on just 2. You're still bringing oxygen in, so you're still doing that process because you're breathing. It's just that you're needing it faster than you're bringing it in. And there's an outer membrane too. That's why we think that these may have been free living, because there's an inner membrane like a cell would have, and then a membrane around it, which the cell that engulfed it would have put around it. Remember, in endocytosis, it builds a membrane around it. Now, the interesting thing about the mitochondria, though, or the thing that's going to become real important to us, is the inner membrane. Notice that it's windy back and forth. That's called Christie when it does that. If you've ever snow skied, you know that's a style of that when you ski down a slope and then back and forth. That's called stem Christie when you do that down a slope. Lots of surface area. Notice it's almost like they took 2 beans of the same size and folded one up and stuck it inside the other one. Lots of surface area. Why surface area? Membranes. Oh my goodness, cells love membranes. Remember I've told you about that? That's why we spent a whole chapter talking about them. We're going to do something with those membranes. You're going to see it's going to be very cool when we get to it in just a second. But you got a lot of that membrane. All right, let me point out a couple of localities. I'll talk about what ATP synthase in just a second. You know, we'll get to that. Don't worry about it. I've already talked about the inner and outer membrane. The inner membrane space, the part that's in the darker brown, all right? So that's not outside, and it's not inside the matrix, the part in the blue. It's that space in between, all right? So you kind of know where we are when I start describing how these places work. Let's see where we are chemically. All right, we're right at the end of glycolysis. So there's glucose, or I'm sorry, there's pyruvate. ATP's already been done, NAD's already been done, all that sort of stuff. We're out in the cytoplasm or cytosol, and here we are. There's a transport protein like y'all wrote about in the exam to me, and that transport protein is going to bring pyruvate into the mitochondria. Now, when you exhale, you give off carbon dioxide. That's some of it right there. Now, what happens inside of the mitochondria, the first step that happens is something we call pyruvate oxidation. Should be obvious to you why the name is pyruvate oxidation. Well, for one thing, pyruvate's involved, and for another thing, oxidation's involved. That is oxidation, right? The way the arrow is going, that's showing you that electrons were lost from something. NAD plus went in, it's oxidized, it gathered electrons, it became reduced, and it became NADH. Now, I haven't talked about where those electrons are go again, right? But we'll get back to those electrons. And then another particle gets involved called coenzyme A, and we make this something called acetyl-CoA. A little sulfur in there, you'll notice. All right? Any questions on that? No ATP was made here, but what did happen is we got the pyruvate into the mitochondria where the big show is going to happen, and we gathered some more electrons. See, if you're not doing lactic acid fermentation as humans, if you're not doing it without oxygen, then the electrons that you got back in glycolysis, right here in this process, they don't go to pyruvate. They actually get shuttled inside the mitochondria too. See, that's what we're saying right here. That's what this arrow, these are the electrons that are being shuttled in. So we're gathering electrons, right? We're harvesting electrons. We did it in glycolysis, and if you got oxygen, you put those in the mitochondria. We've done it here. Now we're gathering electrons. Now I want you to go back for just a second and remember where those electrons come from. They come from food. So these aren't just your everyday run-of-the-mill electrons. These are electrons that have spunk. They have energy. They have octane. They can do work. They have energy because when they were made back in a plant, energy from the sun, right, was converted into chemical energy, electron energy. Remember how many chapters ago we talked about how electrons move between shells? You shine like glow-in-the-dark stuff. The electrons go out. It's all about energy. Electrons are all about energy. That's the whole basis behind quantum mechanics and quantum physics and all that sort of stuff. All right, so this is pyruvate oxidation. So we are done with that. We're right here. So right here, what goes into the citric acid cycle is this stuff right here. Okay? All right, now in the citric acid cycle, first thing, let's do the easy part. The ATP is made through substrate level. Same way I showed you from before, where there's an enzyme and a substrate, enzyme and a product, ATP is made, ATP goes off and does its stuff. Citric acid cycle is really very simple, isn't it? I had professors in high school and college that would make you memorize the names of all these in order, and I always thought to myself, I have no idea what I'm saying. It's like a parrot. Oxaloacetate is turned into citrate, citrate into isocitrate. Isocitrate goes into alpha-ketoglutarate. I always told myself if I taught this to anybody, especially, you know, you guys, at this particular point, it's not the chemistry that's important. I wasn't going to make you memorize it. But there is a couple things I want you to notice. There's this acetyl-CoA. All right. I want you to notice that, number one, this is a cycle. That's why it's called the citric acid cycle. Yes, it does have something to do with citric acid, but no, citrate is made, for example. All right? But the cycle part is what I want you to concentrate on. Mother Nature does this in more places than one. Notice the efficiency of something like this. Notice the brilliance of something like this. What we're saying in a cycle is citrate is turned into number 2, number 3, number 4. Don't worry about all the ending. I'll get those in a second. 5, 6, 7, 8. What you end up with after all those reactions, after what, 8 steps of reactions, is oxaloacetate. Now, oxaloacetate combines with acetyl-CoA from the pyruvate to make citrate. As long as you got acetyl-CoA, you can drive the citric acid cycle. And you're going to have acetyl-CoA as long as you have pyruvate. And you're going to have pyruvate as long as you have glucose. And we're going to have glucose as long as we have plants. All right? So the acetyl-CoA comes from your digestion. This citric acid cycle is sitting there waiting. It's got a substrate called oxaloacetate. There's an enzyme involved in all this. The CoA-SH going off, don't worry about that. That's part of the chemistry. But that enzyme is going to turn oxaloacetate into citrate. And a little bit of water is lost, and we make this other stuff. Now, so you understand the cycle part. Now look what's going on here, here, here. There's the one we haven't talked about yet, but still the same thing, and there. What's going on there? What is the chemical name for what those arrows are indicating? What is NAD plus doing to isocitrate to turn it into alpha-ketoglutarate? What is it chemically doing to it? Or it's also happening down in step 4. FAD is doing it in step 6. And of course, then in step 8, it's also doing it. What is it doing? Ryan, what's happening to isocitrate? Yeah, it's taking electrons off, right? Notice it's going, all of them are taking, FAD, same way, FADH2 is reduced. What we're doing here in the citric acid cycle is we're going through the cycle of reactions. That's nice because you always end up with what you need to keep going, kind of like a perpetual motion machine. But really, one of the big important things that's going on is the energy harvesting. We're gathering not just everyday electrons, though, right? Energy-rich electrons, supposed to be an arrow. We're gathering a whole bunch of energy-rich electrons. That's a big gathering of electrons. So that gathering electrons is something we saw in glycolysis, pyruvate oxidation had occurred. Here, a bunch of them. We must have something real important to do with those. Now, we haven't talked about it yet, but we are in just a second. I do, before I leave, want to show you and remind you that you do make 2 ATPs. It's sort of a gear within a gear kind of thing. First, you actually make GTP, which is just guanine instead of adenine, and then you make ATP. Okay, so you do get a couple of ATPs from the citric acid cycle because of the fact that you split the sugar. Remember, the sugar gets split. They both get converted into G3P, but it's been split. So if you have one glucose, you're going to end up with 2 G3Ps. Each of those goes through the cycle. Each of those makes an ATP. All right, to get ATP, it's a cycle. What in the heck is the deal with the electrons? That's the last part of the story. Yeah, guanine. Yeah, adenine, guanine, cytosine, thymine. It's just one of the other nitrogen spaces. All right, so we're at the end of the story. We're actually following here. Is that the molecule that gives you more? Oh, guanine. I don't know more. I think it's purported to be an energy-releasing molecule. Oh, you know what? They missed one. There's electrons being gathered here, too. Yeah, I know what you're talking about, Robert. I've heard of, but I don't know as far as how it's touted in terms of how much energy you get out versus caffeine. I always wanted to study what the world would be like without caffeine. I mean, I'm not a huge coffee drinker. I try to actually limit how much I drink, but I just think in general, people in the world. You guys all ready to follow those electrons? So I've drawn them with blue arrows. Those are electrons that have been gathered, glycolysis. Remember, in glycolysis, see, this is oxidative. See, oxidative phosphorylation involves oxygen. So those electrons from glycolysis, they're going to go over here because you have oxygen. If you didn't have oxygen, none of this happens, and these electrons go back onto pyruvate or onto acetaldehyde, depending on which are, right? But we do have oxygen, so we make a lot of ATPs. I have the numbers later. We'll get to the numbers in just a second. So far, we've made 2 in glycolysis, 2 in the citric acid. So far, we've got 4. So I'm going to show you how important the mitochondria is by doing the numbers. All right. What are we going to do with those electrons? Okay, here's what we're going to do with them. They're right there. The electrons are going to be shuttled into something we call an electron transport chain. Now, I know this looks real intimidating. All those letters are names for proteins. Site C1, site, I think site is cytochrome. I don't care about the names. What I want you to notice is this looks a lot, I'm not going to go back to that slide of the stairs that I showed you earlier, right? So NADH and FADH2. FADH comes in a little different spot, but they're both looking a little yellow. They're dumping off electrons. They're dumping off high energy electrons. They're dumping off energy, all right, they're dumping off electrons that you can do work with. All right, so as those electrons are traveling through from FMN to FES to Q to blah, 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 to what's the last one? Site. I need new glasses. A3. Those electrons, look on the y-axis, on the dependent variable, the free energy is dropping. Energy is being released. If there's 40 whatchamajiggets, kcals per mole, yeah, 40 kcals per mole of energy at the start, and we're down around 20, then something got released. Now, where did that energy go? You should know. You better know. It's this energy. That energy is the energy that's doing this. I'm not going to draw the other part. It's sticking a phosphate onto that. This is no different than the physical staircase I showed you below. You could take those electrons and put them on hydrogen all at once, get an explosive release. Do we put them on hydrogens here? Yeah. Look down here at the very bottom. Remember I said oxygen has sort of an anticlimactic role. You breathe. Those hydrogens and electrons that were split, I don't want to go back to that slide. Remember, we said the difference is the oxygen goes off and the hydrogens and the electrons split. We take the electrons and we run them through this series of particles like those little wheels, releasing the energy, a little bit of time to make ATP. And finally, at the very end, oxygen comes in and says, you know what? I'm such a pig for electrons. I'll take electrons that have no energy. I'll take those that have very little energy. And so we say, in kind of an anticlimactic way, that oxygen is the final electron acceptor in aerobic respiration, oxygen. That's what we call aerobic. It's the final place the electrons go. It provides a place, just like NAD and NADH that we talked about in glycolysis. If you don't have NAD+, you can't make NADH. If you don't have oxygen, those electrons that are coming down the stairs have no place to go. That's why we say oxygen is the final electron acceptor. And at the very end, the stuff I've circled with blue is when the reunite occurs. The electrons, right? See right there, originally from NADH or FADH2, originally from food, right? Those electrons with low energy get reunited with their hydrogen. You could do the hydrogen ion, the proton, the electron, the oxygen, and there's your metabolic water. So what I mean by oxygen having an anticlimactic role, it's just like it doesn't really get involved until right at the end. So now, if you think about it, it's very simple. If you're in anaerobic conditions, so you don't have any of this, all of this backs up, all of it. But those electrons still have a place to go, or at least they have to have a place to go, and they either go on to pyruvate in animals, or they go on to acetaldehyde after pyruvate if you're talking about a yeast cell. See what I mean? That's how the whole connection works between aerobic and anaerobic. I think we have just enough time at least to get into the next part. So let's dissect this. The word is dissect, by the way. It's not dissect. All right, so let's dissect this part, this whole passing of the electrons. Oh, this is such a cool, because it involves all these things you know. Now, this picture is not in your book, but I know you know this. There's the phospholipids and a bilayer. This is that inner membrane, the cristae. See, this whole machinery that you see that I'm going to explain to you in just a second, this whole machinery, there's thousands of them along that cristae. That's why it's all windy. So you can have a lot of these going on. So we're in that membrane. So this is the beauty of membrane. So the part in the orange out here, this would be the inner membrane space. And then the part in the yellow, this would be the matrix. Maybe I should write inter-membrane. All right, take a look at this picture. Follow the red arrows first. Here's what we're doing. We're pumping hydrogens across the membrane. We're moving hydrogens across the membrane. We're actually moving hydrogens across the membrane using active transport. Okay? So think about that for a second. If we're moving things against the gradient, if we're using active transport, we're going to need a source of energy. Now, if you look through here, you do see energy, but this is energy that's being formed. This is ATP. It's being formed at the end. But look at the very beginning. Carrying electrons from food. NADH is, and so is FADH2. Don't confuse yourself. This is the same as the previous picture. We're just looking at more in terms of the way it really looks in life. So where does the energy come for those three pumps? There's actually three of them. They're numbered 1, 3, and 4 right there. What are these things in between? They're things that help move the electron. The electron is the part moving in the yellow. But where does that energy come from? The electrons from food have it. Food has energy. It got it from the sun. So that's the energy that's used to activate these pumps, these proton complex of electron carriers, and there's 3 pumps. So this is the same picture as before. It's just that, no, 1, there's a pump, 3, and 4. So we're using the energy from the electrons to pump protons across the membrane. You might not need, why would you do that? You're creating a disequilibrium. It's active transport. You're creating a disequilibrium. You're making Mother Nature uncomfortable, quote unquote. You roll a rock uphill, Mother Nature wants it at the bottom of the hill. That's why gravity happens naturally. Diffusion, all of that. Now, think for just a second. What would happen if we're doing active transport? Then this is exactly what's going to happen. What would happen when we have a bunch of protons out here and just a few inside? Mother Nature is going to want to fix that, isn't she? Here's where living systems are so clever. We take advantage of that. We say, oh, you're uncomfortable, Mother Nature. I see. You'd love those protons to go back across the membrane. For one thing, I mentioned a couple times how important membranes are. Look at this. You can't get more on one side of something unless there's something to get more of something on the other side. You got to have a membrane to separate something. So that's why cells are all about the membranes. But back to the diffusion thing. Oh, you can't go through the pump. it's not 100% efficient. Some of those protons in the inner membrane space do actually kind of leak through the phospholipid bilayer. Nothing's perfect, but most of those protons go through a particle at the end that we call ATP synthase. You're right, it is an enzyme. It ends in ASE. Remember I told you sometimes an enzyme will even tell you what it does? So guess what ATP synthase does? Every time a proton flows through it, it's almost like a turnstile. I'm going to go to the next picture. We engage in a process called chemiosmosis. Now, it doesn't involve water. It involves a balance. It's really a fancy word for diffusion that we get work out of. You see, you set up the disequilibrium. The protons want to flow back to equalize, and Mother Nature, living systems say, okay, but you got to go through here. It's like us building a dam to focus water, right? So let me look at a better picture of ATP synthase. Here's the same thing. There it is on the membrane. As the protons are in the gray circles, as the protons flow through ATP synthase, there's a rotor, there's a stator. It's a little motor, man. People into nanotechnology, they love this stuff. Like it's a little, look, it spins. And every time it spins, a little catalytic knob at the bottom, active sites become open. And those active sites are active sites that allow ATP, ADP, and a phosphate to bind. And then ATP is released. So this is an enzymatic reaction. The substrate is the ADP and the phosphate. The ATP is the product. The enzyme is the ATP synthase. Isn't that cool? There's some videos that I actually put on Blackboard that show you little proton flows in, spins around, goes out the other side, and that it's like water going through a dam turning a turbine. There's no water here. The water is the protons. So now you know why we made a disequilibrium, right? We made a disequilibrium in order to get that ATP made. Now, if you're thinking, well, so it's kind of energy in to make energy. All right, yes, but you do get more energy out from the ATP than you brought in from the electrons. You have to, or else this process wouldn't be favored, right? So you concentrate the energy more. It's never 100%, but you're going to want to make sure it's definitely efficient. All right, isn't that cool? Little protons flowing through, turning this thing around, making energy. And again, like I said, that's a pretty good number too. So there we are. 2 in glycolysis. Remember, you actually make 4, but you only net 2. We said 2 in the citric acid cycle, and look at all these guys. All of these electrons coming together, all get dumped into the end. This oxidative phosphorylation is a fancy word for just saying this. When you stick a phosphate onto something, I'm typing. So if you wonder why you're not seeing anything. Yeah, when you stick a phosphate onto something, you phosphorylize it. In this case, you're using oxygen, right? So when you get like, again, it's variable because nothing's 100%, but about 26 to 28 ATP. So think about this. Aerobic respiration, from one glucose, you're going to get about 30 to 32 ATPs. With aerobic. Without oxygen, you only get 2. Well, we do it. Yeah, David. I mean, is there a way that we could tap into the cell? You have to talk to molecular biologists. I'm sure there's something they're working in, but it may not be something that we quite have the technology or the knowledge to do yet, if that's what you're asking. I mean, we harvest it as food. Well, that would be photosynthesis we would have to do. You can't create energy. We're never going to be able, you can't invent something to create it, but we would be harvesting energy, you had to harvest energy from the sun. And artificial photosynthesis, I talked about in class. I said that there are some people working on that, but it's not quite as efficient as we would like. It's like fusion, you know. They can get nuclear fusion to work every once in a while, but it doesn't occur on a continuous basis, that's for sure. Yeah. Well, I mean, yes, that's where it comes from. I mean, I guarantee you're not the only one that's looking at this kind of stuff. I guarantee there are people that with money looking at that. Because you would have the world, oh my God. I mean, if you could do artificial photosynthesis, all you need to do photosynthesis is the sun, hydrogen, and oxygen. Well, you need carbon too, but that's easy enough. Well, I'm sorry, carbon dioxide in water, not oxygen. That's what I meant to say. All right, so there are obviously other places by which you get energy from, not just carbohydrates. Carbohydrates in this picture, they start at the beginning, if you will, but like fatty acids, amino acids, you're not supposed to really get energy from proteins. If you get too much energy from proteins, one of the byproducts is ammonia, and that causes a problem. And some other amino acids. So it's sort of like on a freeway, right? There are various different places that other foods can enter. So that big complex picture I showed you at the beginning, right? They all are going to eventually funnel themselves. Most all of them, I think, eventually funnel themselves into the citric acid cycle, one way or the other. So look at down below, I kind of show you, that's the citric acid cycle, and these are a bunch of amino acids all around. So many different amino acids could somewhere funnel into there. And the arrows with the glucose going back and forth means your body actually uses glucose and stores glucose. So it's a balance, right? Well, perfect. I got one more slide to go, and we were just talking about balance, so this should work very nicely. Everything is regulated. And as you know, feedback inhibition of an enzyme is the best way to do it. So look at the one on the left. When you make an excess of ATP, you remember feedback inhibition, it's all about the excess. Yeah, that's a long time because usually it's a few milliseconds. But if you got too much ATP, which is like having too much milk, ATP actually itself, what better to be the feedback mechanism regulator than the thing you're making? The final end product, ATP, inhibits one of those enzymes. Don't worry about the name, but look, we're way up at the beginning of a glycolysis. Remember, you're typically going to want to inhibit an enzyme early up. So phosphofructokinase gets inhibited by excess ATP. And there's a variety of places you get ATP. That's why the arrows are there. In the citric acid cycle, a material called citrate, remember that was the first step that was made, is where we get the name from. An excess of that will also help down the enzyme. So you got a break in, and you got two breaks, if you will. Now, one thing that we didn't talk about, but it kind of makes sense, if your body gets low on energy, like if you get a lot of AMP is ADP. See, ADP and ATP are usually going to be recycled because you generally don't want to get to AMP. Then the energy level is a lot less. So if you have a lot of AMP, then you do the opposite to the enzyme. You actually stimulate the enzyme. See, an inhibitor could change the shape of an enzyme and cause it to be less efficient, but it could change the shape of an enzyme and make it more efficient or make it more attractive to these substrates. Right? Everything's got to be regulated. Right? This is just an example of regulation that we've talked about all along. And as I said, feedback is going to be a way that you'll see a lot. What better way to regulate something than the excess final end product, right? So you don't make too much of something, you make just the right amount. Homeostasis. All right, that's it. I'm going to stop the recording here.